So thanks all for joining. I'm going to talk a little bit about hemlock woolly adelgid today. Yeah. So before we start, I always like to have sort of acknowledgement slide of the folks we have working in the lab. This photo is actually from 2 years ago, but it sort of highlights all the folks we've had working on hemlock woolly adelgid over the past couple of years. And actually, when I was putting this photo to get this presentation together today, I realized that in the background here, just change slides. There we go. We actually have Tom, who wasn't working with us at the time, but just happened to be walking by at the lunch we were having, and so we caught him in the picture. So Tom is actually working with us right now, and he's in the lab photo. So always photobomb is what that. I think that's Vince from. Isn't that Vince from ISC as well? Yeah. Okay. Yeah. So, yeah. All right. Okay. So I'm going to talk a little bit about hemlock woolly adelgid. Okay. So this is the hemlock woolly adelgid. The photo on the right-hand side that's the insect. So it's super tiny. It's related to aphids and scales. And what it does is when it lands on the tree, or when it's born on the tree, it crawls around, inserts its mouth parts, and it won't move again for the rest of its life. And because of that, it has to protect itself, so it makes its white woolly coating. So hemlock woolly adelgid. We'll just call it HWA. Photo on the right is a blow-up picture that I think it's Victoria's, or whose is that? Yeah. One of Victoria's photos through the microscope showing the actual crawler stage. So turn out the crawler stage, one of the nymphal stages of HWA, as it's about to sort of set up on a twig. So super tiny. Those are hemlock needles you can see there. That's just an idea of how small the insect is. So we're dealing with quite a small insect. Has a super complicated life cycle. It has 2 asexual generations during the year. It also has a sexual generation that has wings, but that sexual generation needs a specific kind of host tree that we don't have in North America, so it's a kind of a dead end. Right around this time of year, normally the insects would still be asleep, but because it's so warm, they're actually starting to wake up. So they start to wake up. They lay some eggs. Those eggs hatch to the crawler stage, which moves around. Those crawlers settle and sort of pass through a succession of nymphal instars before they become adults. And then those adults lay eggs. Those eggs hatch to another sort of longer crawler stage that goes through until the fall, forms some nymphal stages, and then they emerge as adults. You can see that sort of the hash marking there on the crawler stage in the cystens generation. That's a summer estivation where the insect actually sort of goes in kind of dormancy, so it kind of hangs out and waits during the summer generation, and then it wakes back up in the fall. For some reason, we call the one generation the cystens generation. For other ones, we call it the Progrediens generation. I don't know why. It's like a taxonomy thing. It just makes it super complicated. This is perhaps the most complicated insect life cycle we've ever worked on, and it's a giant pain in the ass. So basically, it's an invasive insect. I'll talk a bit about the introduction and spread in a bit, but kills eastern hemlock trees in 3 to 6 years. So it's a rather significant mortality factor for hemlock trees. And that's just dead and dying trees there from the Appalachians. So talk a bit about the introduction and spread into North America. Was initially detected in Virginia in an arboretum in the 1950s, was probably brought over on horticulture stock from Japan. And it slowly spread. It didn't really get on the radar until the 1990s, when it really started to become apparent in places like Connecticut and where a lot of the initial work was done. It was found in upstate New York in the early 2000s, and then in Nova Scotia we found it in 2017. And then I'll talk a little bit about Ontario here in a bit. A lot of the dispersal is either by movement of wooden plants, which is kind of your standard invasive species thing. You get hemlock woolly adelgid on a tree, move that tree around, it gets to a new place. Dispersal within stands by vertebrates. There's this crawler stage that it produces tons of insects, and these get moved around by things like deer and humans, and then by birds. And a lot of the northward spread, we think, is by the movement of the crawler stage on birds. That's that generation that I've circled there. When that's present on trees, it corresponds to spring migration. So as you have birds coming up from the south, they're landing on hemlock trees, they're picking up crawlers, flying to new trees, landing, those crawlers come off. And because the insect is parthenogenic, it's asexual, doesn't need to mate to reproduce. So the minute it's introduced, it can, in theory, establish a new population, even from just one insect. Where hemlock woolly adelgid is neat is that on the west coast of North America, it is a native species. So it's this really weird, unique invasive species thing where west coast, it's a native species, east coast, it's an invasive species. And that's because in historical time, in geological time, Western North America and Asia were connected. There was a land bridge, climate was warmer, hemlock spread across that whole Pacific rim. And during that time, hemlock woolly adelgid spread from Asia to North America. Climate changes, land bridge disappears, hemlock disappears, but you have this remnant hemlock, and you have remnant hemlock woolly adelgid populations on the west coast. So what I'm showing here is a molecular phylogeny by Nathan Havel, where they collected insects from all over Asia, hemlock woolly adelgid from all over Asia, and then looked at the evolutionary relationships. And what it tells us is that our populations in eastern North America are most closely related to those from Japan, right? Which corresponds to the invasion, we think the invasion history was. Those populations from Western North America are distinct, right? So the insects we have in eastern North America are not from Western North America, and they're distinct from the populations in Asia. So these are different populations. And because of that, in Western North America, hemlock woolly adelgid is not a pest. It has it's co-evolved with its host tree, which can defend itself, and it has a full suite of natural enemies that keep it in check. That doesn't exist in Eastern North America. So in the East, it's an invasive species. In the West, it's not. So just to summarize, that's the range on the West Coast, and that's actually all from FIDS data. So we've been collecting hemlock woolly adelgid going back to actually since before there was a Forest Service. One of the first pests recorded from Stanley Park is hemlock woolly adelgid. But on the East Coast, invasive species, and that's all of the records for hemlock woolly adelgid in Nova Scotia and Ontario. So this is just kind of a history of the hemlock woolly adelgid infestation in Ontario. So first population is 2012 in Etobicoke. That was an introduction of horticulture stock. Jeff Fidgen, Jean Turgeon, our colleagues in the CFIA and the MNR responded to that one and eradicated it. So all the infested trees removed, burned, and then there was an intensive survey around those homes, and no hemlock woolly adelgid was found. 2 years later in Niagara Falls, one tree in the Niagara Gorge, same thing, cut down, burned, got rid of, survey, no insects. That was the state until 2019, when it was found again in Niagara Falls on a couple of trees, and in Waynflete County, right, in a large piece of property called the Harold Mitchell Nature Reserve, which is like a large hemlock stand. At that point, that infestation was quite well advanced. It had been there a long time. And since that time, we found new infestations every year in Ontario summer, except for 2020, when there wasn't a lot of survey going for kind of obvious reasons. This one in Grafton, we're somewhat proud of. Megan and a crew from Guelph actually found this. They were in a stand looking for hemlock trees for one of our other projects with a colleague at Guelph. And Megan called at like 10 o'clock in the morning and says, "I think we have hemlock woolly adelgid," and they did. So it's a rather uncomfortable call to our colleagues in the Food Inspection Agency to tell them to send somebody out to find this. And I'll show you, Grafton is well removed, and hemlock woolly adelgid wasn't supposed to be there. So this has turned into a research site for us, but it was a fun story and to find hemlock woolly adelgid there, and it's precipitated a lot of the work that we have done now because it is so widespread. 2023, we found it. It was found in Hamilton at the Royal Botanical Gardens, Halden County, which we'll talk about in a little bit, and then Lincoln, which is our friends at the MNR. There we go. So, what's the risk? Hemlock occurs on about greater than 1 million hectares in Ontario, and this is just the sort of known hemlock stands from Ontario's inventory and a product put together by Kathleen Ryan, assembling all sort of hemlock inventories from municipalities and conservation authorities in Southern Ontario. So 1% of those growing stock, it's a really significant component of riparian and old growth forest communities. So especially in Southern Ontario, if you find hemlock, you're probably looking at an old growth remnant forest, something highly significant, important to conservation authorities or landowners. There is a component of it in the forest industry. It's used for like harvested for saw logs, pulp, fuel wood. Mennonites apparently will use a lot of hemlock for barns and like sheds and this kind of thing. So it is harvested, it is moved around. It's not sort of a major forestry species, but between the impacts on forestry and the impacts on ecosystems is why we're worried about hemlock woolly adelgid. So what I'm going to talk about for the rest is just the research that we've been doing sort of here at the building and over the past sort of 10-ish years, going back to about 2012. We were really fortunate sort of in those first finds in 2012 is that we had Jeff Fidgen working here. Jeff's now at Atlantic. Jeff did some work in Scott Salem's lab in Virginia, right in the heart of hemlock woolly adelgid territory in the States. Had a lot of experience with it when it was found. And he started working with Jean on developing sampling tools. So Jean and Jeff and some other folks and myself worked on developing sampling tools and tactics for hemlock woolly adelgid. I had Carolyn Emerson working with me for a while, so we had Carolyn write a hemlock woolly adelgid management plan for Canada that sort of influenced a lot of what's been done in other places. We developed this biological control framework for Nova Scotia. We have this hemlock woolly adelgid technical advisory committee that we work with the Invasive Species Centre and Food Inspection Agency and MNR. And then Kathleen Ryan has had a hemlock woolly adelgid working group. And these last two are sort of what's influenced kind of our research direction, the questions they have had about what to do, what HWA has influenced what we've been trying to do. And so some of these products are a response to that. Since about 2019, we've been looking at sort of improving detection and trapping tools. This is some work that Victoria's been doing, and we worked with Dennis on some modeling stuff. That's the optimizing strategy for allocating resources between different agencies. We were looking at overwinter survival of the insect in Ontario, Nova Scotia. We didn't do that this winter because it would have been really boring. We did it last winter, and I'll show that in a bit. And we've had a student with Lakehead and now with the University of Toronto looking at species distribution modeling for hemlock woolly adelgid. And then with Bill Parker and a bunch of folks in the MNR, we looked at all the silviculture research from the States and through the lens of Ontario silvicultural guidelines for hemlock, put together a paper and some other knowledge transfer products for FMLs and landowners in Southern Ontario to manage their hemlock, to kind of increase resilience to hemlock woolly adelgid, and then what to do if they get it. So our research response, and these buckets we sort of chunk these research into different buckets based on whether it's assessing risk, mitigating that risk, or developing sort of standard resilience. We've been looking at sort of revising hemlock inventories for Southern Ontario. That's a project with Ben DeVries at Guelph. We're looking at Cameron's species distribution modeling. We're looking at phenology, some other risk mitigation stuff that we've been working on that I'll talk about. And then there's a management guide for hemlock woolly adelgid that Victoria's been working on with the Invasive Species Centre that should be out soon-ish. Yeah, Doris is nodding. April, we hope. That's supposed to be an online evergreen product for people to look at to sort of best available science, best available advice for hemlock. So I'm going to talk about just briefly four of the projects that we have going on. The first one has been the big one that's been kind of all the work in the lab kind of for certainly last summer and a chunk of this summer will be looking at phenology of HWA in Ontario. So what we did is went to two of the stands in Southern Ontario, one of the finds at Short Hills, which is in Pelham, and then the Grafton find. And we sampled these populations every 2 weeks for the first part of spring, kind of through the assistance generation. And then later on in the summer for the progredians, when it's a bit slower, we sampled every week, every 2 weeks. And that sampling consisted of cutting a couple branches, zipping them up into some double bag, secure transport because we're moving from an infested area up to the Sioux, brought them into IPQL under quarantine, and then counted everything on these branches. And when I say we, it wasn't me, it was Victoria and Madeline Swin, who was our summer student, and Megan and some other folks we had working in the lab, did all this counting. So ton of counting. And what we were trying to do then is basically fit this model from Patrick Tobin and Richard Turcott, which is basically just an exponential growth model, just trying to figure out like at what time of year in terms of degree days are these different critical stages appearing on the landscape. And we need this if we want to do even like real basic stuff like apply insecticides or apply biological control agents, or just know like what's the population doing now so we can time our own sampling and things like trapping and whatnot. So when we did that, fit those, do all that counting, fit the model, you get graphs like this, and you can sort of extrapolate from the accumulated degree days roughly when egg lay begins in these sites. So egg lay last year was about anywhere from April 21st to May 6th. Egg hatch, which is the orange line, about the 18th to 30th of May. And then you notice there's some spots in here that we didn't model, and I'll explain that in a moment. And then we have Sistans estivation, which is that summer dormancy happening sort of mid-July. We missed the crawler stage in 2024 because of a series of unfortunate events. You may recall there was a strike in the public service when a bunch of our folks were not in the building. So what happened during that is we were still getting samples, we were collecting them, we were still getting them from M&R, but we had a big backlog of stuff that was sitting down in the cooler. And by the time we got processed through the backlog, we realized that the samples we were getting from some of the sites weren't of the right stage, and they weren't the trees weren't in the right condition. So basically we missed this. And by the time we realized that and said, could you collect from some different trees, please? We had missed that stage of the insect. So this year we will attempt to fix that and get these other stages that we missed this year. So that's one of those challenges of field work. So what we did with those data is just try to predict, what's the rough calendar days might you expect to see these Brigadians eggs or hatch or cystens appearing on the landscape, just based on available climate data. So what I did is I for those sites, I pulled the last 5 years of climate data and just translated degree days from those years into calendar day, and then just did the mean calendar day. And so Grafton, Ontario, and Short Hills are our 2 sample sites. I was also interested for the Sioux, partly because we live here. We're at the northern edge of the range of hemlock, and hemlock is actually as far north as Sleeping Bear Dunes National Park or State Park in Michigan. So there's a risk that hemlock woolly adelgid could get here. I was curious, could it develop, right? So we don't have it here, but I just modeled it anyway. And so what we found, there's a really big variation between sites in the Niagara, which is Short Hills, and the North Shore of Lake Ontario, which is cooler, right? So over a very short geographic distance, there's a huge range of developmental timing depending on where you are and what the local climate is. And looking at the Sioux, it's even possible that the insect could develop here. It's certainly a lot later, like it's almost a month later than at Short Hills in the Niagara. But extrapolating this out through the development time of the insect through our climate, you could grow a population of hemlock woolly adelgid here and have it survive. So that's sort of it's one estimation of risk for this area. So change gears a little bit and talk about overwintering survival. Folks might remember that at the beginning of February last year, there was one of these kind of polar vortex events. It wasn't actually a polar vortex, but it was this rapid outflow of cold air from the Arctic, sweeping across eastern North America and plunged temperatures, really significantly cold over the night of February 3rd and 4th. I'm Facebook friends with a guy named Jan Belanger at Laurentian Forestry Centre, and Jan, for fun, will post climate prediction models for the future on his Facebook page, and he'll talk about them mostly in French, but but what he did is he was posting stuff about a week before this from these models, these European climate models that I don't understand, saying this thing was going to happen. I'm like, oh, is this really going to happen? He's like, yeah, no, it's totally going to happen. So what we did is we phoned up Jeff Fidgen in Atlantic. I said, could you get some samples from the Maritimes before and then again after, so we can see what is the effect of this big cold event on HWA. And he's like, yeah, sure, no, we can do that. And so then we phoned up some folks in the CFIA and Columbia checked down in Guelph and said, could you get some samples from Wainfleet? She said, yeah, sure, okay. So between them, we got these samples like right before, and then we got them right after, and we were able to assess survival. And so when you do that, you get a curve like this. So you get a figure like this. So on the right-hand side, these are temperature traces from the 1st of January 2023 through to about March. And then the big spike, that's the night of February 3rd, 4th at Annapolis Royal and Mersey River in Nova Scotia, Grafton, and Wainfleet, which are our sites in Southern Ontario. And then on the left, you see got down to -25, -23, -26, -17 at Wainfleet. On the left-hand side of the survival curves, or the survival estimates of hemlock woolly adelgid right before, so that's the white, and then right after, and then the lines connect the same trees. So we sampled the same trees before and after. And what we found is there was this really significant mortality at Annapolis Royal, Mersey, and there's big mortality at Grafton, but we weren't able to get a sample right before, but not at Wainfleet. And that correlates with these temperature drops below -20, -23, right? You get big mortality. But what's interesting and what's important for us is that none of those in Nova Scotia and even at Grafton were significant enough to wipe out the population. So that's really significant mortality that dropped it sort of like the survivorship to 10 or 20%. But because the insect is parthenogenic, it can rebound its populations really quickly. So we wrote this up and we just actually had it published like last Monday current research in insect science. So that's out there for folks to read. And this is actually the first assessment of the insect at the northern edge of its range. So it was a fun little project, and it all started because of like Facebook social media stuff. So it's not all bad. Okay, so talk a bit about range expansion. So one of the questions we have for hemlock woolly adelgid in Ontario is like, how far can it spread? And this is like a question that I've had for a really long time. And like so the green is the range of hemlock, and those are our sites. But like, where could it go? I have a new colleague at Lakehead University in Thunder Bay. His name's Sungil Lee. Sungil's from Alberta, and he had a master's. He had a undergraduate student who's interested in doing some species distribution modeling using iNaturalist data for hemlock woolly adelgid, just like out of the blue. He says, "Are you interested in this?" I'm like, "I'm totally interested in that." So Cameron basically built the species distribution model for hemlock woolly adelgid for Eastern North America, which is what I'm going to show you. But he also did it for Western North America and Asia, using all iNat data and some of our old FIDS data to build these species distribution models. And when you do that for hemlock woolly adelgid, all the assumptions of species distribution modeling, taken into account, and that these are kind of hypotheses that would need to be tested. So what you have is all the red stuff is known locations of HWA based on iNaturalist, and then the gray black shading is where Cameron's model estimates that hemlock woolly adelgid could go in Ontario. And so all of that part of southern Ontario, north shore of Lake Ontario, into kind of the Muskokas and Kawartha areas, which is somewhat of a risk, and then even up along the north shore of the Georgian Bay. So that's a little bit further than some of the other models predicted, and this is based on kind of overwintering temperature models that he had. And then the next thing Cameron did is he pushed that through some climate forcing scenarios, basically low emissions, high emissions, mid emissions. Do we all start driving Teslas, all this other kind of stuff. And if you do that, you get all these crazy species distribution models. And the ones I'd have you look at are just in the yellow box there. And basically, even under like a low emission scenario and a mid emission scenario in the near future, so out to like 2060, what it basically predicts is that hemlock woolly adelgid could fill the range of hemlock in Ontario. Right? So that's kind of like the potential risk under kind of a kind of business as usual type scenario. It gets really crazy if you do sort of high emission, mid emission scenarios. It like pushes it up to James Bay, well outside the range of hemlock. So these are hypotheses that we could test, right? But in terms of assessing risk and probability of occurrence, this is kind of what Cameron's work tells us. So the last thing we'll talk about is some of our eDNA detection work with Invasive Species Centre and the Food Inspection Agency. So if we kind of know insect can survive in Ontario, it's being introduced, potential for it to expand its range, we should probably try surveying for it. The CFIA does that. There's only so many of them, and they can only reach so many spots. A lot of the hemlock trees in southern Ontario are privately owned or owned by conservation authorities, so it's difficult to get in and survey those. So we sort of became aware of this this new trap design from Charlotte Partridge, who's a prof at Grand Valley State in Michigan, and it's made with a 3D printer. So that's the main body of the trap there. It's kind of a two-piece thing. And then in between the two pieces, you put microscope slides that you have coated in Vaseline. So you dip them in Vaseline, put them in this trap, the top goes on, and then you mount it to a stick underneath a hemlock tree. And what happens is that crawler stage falls out of the tree during sort of that, when it's present, and it gets caught on the microscope slides. And then you can take those slides back to the lab and you can look at them underneath the microscope and just see do you have crawlers. And if you're not sure, you can even scrape them, then you can scrape the Vaseline off, extract the kind of fluid from that, send it off to a lab and assess it for hemlock woolly adelgid DNA, right? So say, do we have eDNA, environmental DNA on these slides? So Charlene developed this in Michigan. She's a social media person. I am sort of a social media person. I'm like, this thing is cool. Can we try this? She's like, yeah, sure. So we tried it in Grafton for summer and it's like worked pretty well. We liked it. And so we partnered with the Invasive Species Centre and the Food Inspection Agency to turn this into a community science project to see if we could detect hemlock woolly adelgid. So what we did is printed a bunch of these traps, coated them with Vaseline, put together these kits, and I'll show you those in a minute. Sent them out to folks in southern Ontario. They come back to the lab and they hear the risk of scraping some of the slides, putting them, inspecting them for hemlock woolly adelgid. and then they get sent off to Aaron Campbell's lab at the Canadian Food Inspection Agency to do the DNA work with primers that were developed by Mark Whitmore's lab. So that's the basic project. And we took our inspiration from budworm, because everything in the Forest Service is inspired by budworm. So for a number of years in Eastern North America, we've had the budworm tracker program running. This is all of our sites in Ontario for last year. So it's a community science project where we send people a trap and send people some lures, and they send us back the bugs, and we use that to get an additional assessments of what budworm looked like. So Bill out the same kind of program for hemlock, woolly adelgid, but just this time in Southern Ontario. So what I'm showing here are in red is all of the sites that got a kit. Is that right? Yeah, all of the red sites got a kit last summer. So there's 50 people that that were sent one of these kits, and the kit is just the trap and the microscope slides and some gloves and a couple falcon tubes and some instructions. People had to supply their own stick because we didn't want to nail a stick. And the blue dots, just those are the other people that volunteered, like they were interested in getting traps. We had a lot more interest than we were able to help to supply. Sent all these kits out. We got basically all of them back. One person had theirs stolen. Somebody put theirs in upside down, like in the slides in upside down, which was unfortunate because I think they may have caught a hemlock woolly adelgid crawler at the edge of one of those slides, but we couldn't tell because it was kind of smooshed. What we did get, which was the most exciting one, at least for us, again, not for our friends at the CFIA, because we had to call them up and say we found another HWA site that you didn't know about, was this one in Haldeman County. So this is one of our budworm tracker, or sorry, one of our hemlock woolly adelgid eDNA folks. They got their trap, sent us back the slides, and when Victoria cracked it open in the lab, she's like, yeah, it was like, it looked like somebody shaking pepper on it, like it was full of crawlers. And so phone up Nicole at CFIA, and they sent out a crew, and sure enough, within about, I think it was within 5 minutes, they'd found hemlock woolly adelgid in the stand, like it was fully infested. And it turns out this is a piece of property that's like the forest extends into two pieces of private property and some conservation authority land, and they've since found it kind of in this general area. So very exciting. Like we kind of wanted it. We did want to find one infestation in the first year just to prove that it works, because we don't find anything, you know. So we lucked out. We've perhaps been a little bit of a victim of our own success here. We sort of planned to go slow with this. We were going to have maybe another 50 traps this year and then talk about what we're going to do next. We've had interest from all the eastern provinces now in setting up their own detection networks. So these traps will be in Nova Scotia and Quebec for sure. That we're going to try and roll out some more traps in southwestern Ontario, so the London area and down there, to pick up, so we can pick up more populations. And then I think they might be in New Brunswick possibly next year. And PEI, yes. They're going to sample underneath the hemlock tree in PEI. I think it's what they have more hemlock. I'm just joking. My grandfather's from PEI. I can make it. It's okay. I can make it. Okay. So if anybody happened to see the presentation at Pest Forum, this was called the Fellowship of the Trap. There was some unfortunate presentation graphics from Olivier in CFIA, photoshopping our heads onto the Lord of the Rings posters. Because it's basically it is a fellowship amongst our three groups. So Invasive Species Center does all our program coordination and product development. So these they do all of our outreach, all of our shipping and handling, dealing with the folks, recruiting and then dealing with those folks. CFIA does all of our site inspections. So if we find it, they're the only one that can actually say yes, it's infested. And then they do the eDNA analysis in Aaron Campbell's lab. And then what we do is, we apply the protocol expertise. My lab and sort of Victoria Derry has been coordinating all this for our lab. We do all the protocol development. We host the kind of scraping of the sealing off of the slides and the setting up of all the slides, and then all the trap processing, and then sort of supplying that research component, right? So how far to put the traps out, and working with Charlene to optimize some of the things. So what we're hoping is that we can sort of continue to do that research component on the trapping, but that we can sort of operationalize and get somebody else to really expand the eDNA trapping network. And it's a really neat method. It builds off of some of the stuff that Jeff Fidgen did, which was just like, Jeff started doing green sticky cards underneath hemlock trees. It worked really well, but with those you get a lot of bycatch. The neat thing about these is you hardly get any bycatch. There's hardly any other insects on these traps. So we had some interest from other folks looking at other trapping technology. So it's a really neat thing. It's turned into kind of a neat project. And so it's kind of what we've been doing. We're going to keep going. We've got some other work with, looking at faking on targets of insecticides, and that we hope to get into, and then completing our phenology work this year. So that's my talk. Hey, Barry.
Hey, Chris, great talk. What's the alternate host for the flying stage? And is it like a Japanese ornamental? Is that what they caught it on in Virginia in 1950?
So Barry's asking what's the alternate stage for the sexual generation. It's something called tiger tail spruce, which is some exotic. Well, sorry, not exotic. Exotic to us. It exists in Asia. And so hemlock adelgid was probably brought in on the Japanese hemlock for planting, and then but we know in Asia that it needs that tiger tail spruce. So tiger tail spruce, it's not very common, and it's actually a regulated plant in Canada. You can't bring it in, you can't have it. So yeah. Jason.
Great talk, Chris. Can you talk a little bit about the trap design? It looks really interesting, and you said it doesn't get a lot of bycatch.
Yeah.
Is that to do with the interesting circle? Can you speak to what's going on?
So I think I probably glossed over the fact, it's made with a 3D printer, right? Which is so Charlotte had a graduate student that designed 3D printed design. So it's meant to be built on a 3D printer. So a lot of the kind of holes and spacing is just to minimize the amount of plastic, right? Which is kind of the cool thing. Normally insect traps, somebody designs them and then they license them to a company who builds them and sells them. And they're really expensive because they're kind of a bespoke product almost. What Charlotte's done by designing this is an open source thing, is she just made the design available on her website. So if you have a 3D printer and you can buy some microscope slides from Amazon, and you have Vaseline, and you know somebody that can do eDNA amplification, you don't need us at all. You can do this on your own. So a lot of that design is just to accommodate that. Her original design was a pie plate, right? And it actually spun, there was a motor and stuff, but you realize it didn't need to spin. So so it's based off of that. That's why it looks the way it looks. Yeah. So because it's not the crawlers are super tiny, and so when they land on the Vaseline, they almost become embedded in it. So for them, it's sticky. For something like a little bit bigger, like a beetle or a fly, they can extract themselves from it. And we run them a little bit longer than Charlene does, but so she only runs them for a week and a half. We let them a little bit longer. And so that's compared to some of the other trapping methods that we use, which is just we'll put bug stick them or bird stick them on them, and that catches everything, right? And including birds, right? So it's getting away from that kind of, and certainly it makes them easier to process. If you've got a sticky card that's full of everything and there's little tiny crawlers, you have to go and look for them. So that's kind of the nice thing about it. So, yeah.
I was wondering, do you have a gut feeling for increasing the soup? How long it's gonna take for us or for the trap to detect it?
How long does it have to be there before it gets picked up in the trap?
Yeah.
That's an interesting question. I don't have a good answer for that one. Some of the other detection methods that Jeff developed, which is ball sampling and putting the sticky cards and then even visual inspections, we know that you can pick it up fairly early if you combine techniques, right? So the ball sampling is really good at getting up at the top of trees. So that's a racquetball with Velcro on it that you use with a ball launch, you shoot it up through the tree. It works really well. It works even better if you stack it with another technique or two. And folks like using it, but they complain that it apparently, it's a lot of work to do this over and over again. And I've sent people to the field who've done it over and over again, and they enjoy it a little bit, I think. Anyway, Allison's here.
It was fun.
Yes.
Cool.
Yeah.
For the first hour.
Yeah. It looks super cool to do, right? But there's so we get some resistance from folks to using it. So that's maybe where the trapping works a little bit better. But the question about at what stage of infestation do you detect it would be cool to get at. But I think you'd have to artificially infest some trees, and I don't know anybody that would let me do that. That's always the trick. Yeah. I guess Amanda, and then Barry.
How far away can you be from the tree?
So Charlene says 300 meters. Yeah, we tried it in Wainfleet, and we had them a little bit farther out than that. We didn't pick it up, but Charlene's research in infested stands in Michigan about 300 meters. So, which isn't bad, right? And then there's the fluff too, and that, so that kind of thing. Barry?
That point down south on Lake Erie, was that near Long Point? And is that on Bird Flyway? Is that how they got there?
So,
Birds coming in
I
there?
think it probably is birds. That site in Haldeman is east of Long Point. But if you look at some of the radar tracking data for birds, what they do is they certainly funnel through the lakes and then they move around, right? They're flying along the lakes before they go north, or they're just moving, right? So what I've wanted somebody to do, who's like an ornithologist and who understands bird tracking, is to kind of build a spatial model using some of this really high resolution radar bird tracking data that exists for, and make that to a phenology model to help us predict, what's the critical point when birds are moving through and picking them up and moving them. Because that would, if we could do that, we could tell CFIA, you need to focus your survey over here, or these landowners need to look more. So, but I don't do that. I'd like somebody to build it. So yeah. If you're online and you know how to do that, I get in touch. Amanda.
Building on that, there's a ton of bird monitoring down in that area because birds do this setting. Did you like bird foot washing just to look and see if they're on there? Like,
I think
a little wash.
so. I think it could. Yeah. Could you like wash the feet of birds? I think you probably could. I think it's been done in the States. There are a couple papers on bird migration. I think it could totally be done. I draw the line at like insects, like when we get to 2 legs, I will help with somebody, but that's birthday, right? So Megan would totally do that. Okay, so if you're an ornithologist and listening, love to talk to you. So, Mark. Yeah. So the question was like, what's the probability, like the likelihood of eradication in Canada? So the first two populations were successfully eradicated. They were kind of special cases. The first one was like definitely on horticulture trees. Somebody bought them from like infested from a nursery. They weren't very old. And the survey around there said that so that you can remove that. That's doable. The one in Niagara was one tree, cut down, burned, and then lots of survey afterwards. So if you know where to go and look, it's almost like that streetlight problem. Like if you know where to go look, right, you can say with some confidence that it's not there. But there's kind of like a growth curve, and if you go past a certain tipping point, you can no longer eradicate, right? And Wainfleet was at that point when it was found, right? And so like we walked in there, we went with CFIA, and you could find it on the little trees just looking around, right? So once you're at that point, eradication ceases to become an option for an insect with the biology like hemlock woolly adelgid. So immediately you're into management, right? You're trying to find it. You're trying to figure out what kind of tactics can you use to treat trees. Can you introduce natural enemies, that kind of thing. So you immediately move into a different kind of pest management. Yeah. Yeah.
Yeah, we want to try to do something.
Yeah, sure. Yeah. Dan would like to meet about economics. That's Dan McKinney, our economist. Okay. Yeah. Oh, okay. So the question is about the native predators on the West Coast. So there's a couple beetles that eat it. There's a couple predaceous flies. So they're predators as larvae, and so as larvae they eat hemlock woolly adelgid. These three, and there's three of those species have been introduced to Eastern North America as biological control agents, and one of them has been introduced to Nova Scotia to start biological control there. So yeah, that's what exists. There's other things that eat them. It was reasonably well surveyed in some parts of BC. There's other places in BC that we'd like to do some more surveying to see what other kind of predators they might have. So yeah. Dan. The insecticide work? Yeah, so John worked for a number of years on the populations in Nova Scotia looking at like insecticide treatments. Because a lot of John's work, we now have when hemlock woolly adelgid was found with one product registered. It's called Imajet. It's imidacloprid, systemic injected into the trees. Bioforest and John worked together, and they eventually registered triazine. So they added hemlock adelgid to the label for triazine. That's the one that was developed here. There's a number of products in the States, and the companies have brought those into Canada under emergency registration. So there's another Imajet product, Imajet 10, which is just sort of additional concentration. Has full registration. And then there's two products, one called Starkle and one called Zytec, which are both neonicotinoids. One's midacloprid, one's dinotefuran. They're both sprayed products, and they're now under emergency registration in Canada, and they're available in Nova Scotia and Ontario until the fall. So yeah, and then the hope is the companies would bring them in, and they're used quite extensively in the States. They work pretty well. The new products are bark sprays. You can go in with a backpack sprayer and just apply them to the bark of the tree. It gets absorbed, and you get pretty good action for a number of years against HWA. Yeah. So they will settle on the new shoots, right? So the tree makes new shoots each year, they're settling on them, and so its populations build up, right? They will settle further back, but that's where you get those. That's where you get that essentially, right? These big populations building up. They start at the top and they sort of work their way down, right? So the tree becomes more and more infested. There's this thing where like they become infested, and then the trees sort of get so stressed out, the insects actually start to die. So they die back quite a bit, and then the tree reflushes, and then they go, oh great, we can eat here again. So they come back a little bit, so you get kind of this double thing, and that's maybe one of the things contributes to the end of the tree. So the populations build up really quick, and because it reproduces asexually, it can go really, really fast. So, yeah, those samples came in last week, and I think Victoria is she actually down there right now looking at them? Or?
I think she's online right now.
Okay.
Yeah.
Yeah. So they're well ahead of where they were last year. So I think like Megan and Victoria were in Grafton almost this time last year, and the text I was getting was, "We wish we brought the snowshoes so they could get in." Right. There was a walk. They were in there last week, and it was a walk.
So it's very different.
Yeah. So, Barry.
I don't know anything about adelgid population dynamics, and I also don't know anything about indigenous versus invasive species in North America in that family. So, do you know much about that? Can you give me a sense, like what's the real risk here? Are there other cases where adelgids have come in and stormed parts of Canada or the U.S.? Like, give me a sense for what's the risk here.
So, I mean, we can estimate sort of the risk a little bit from the States for adelgid. Like there's been this long history, and certainly there's been pretty significant effects on hemlock. The only analog I'm really familiar with is balsam woolly adelgid. That's another invasive species. CFS did a lot of work on balsam woolly adelgid. He's going back to the 50s, I think, 40s, 50s, in the Maritimes and up into the Newfoundland. It's now kind of everywhere, right? It's out in Utah and all those other kind of places. So it's spread. It's almost one of these invasives that was a big problem, and it kind of went away. And because it went away, nobody really paid attention to it, and now it's kind of back in other places. So these things are a pain to work on, right? So they don't attract as much attention as something that's a little bit more tractable, I guess, or sexy. I mean, so yeah. Cool. All right.
Here.
Great. Thank you. Thanks for coming.